What's up?
Not much. How are you?
I'm okay. I did couple cars today, couple cars passages. Um, I like fell down a little rabbit hole. I did like blind review of like seven passages. I was like, you know what? I'm feeling it. And then I just did like four more passages.
Look at you. Yeah. So I was gonna also try to say something to like help you like brainwash yourself to like finding the stuff that you read about interesting. Um, but all right, cool. How did they go?
So I got, well, for three, I got 87%, which I was happy about. And then I did one other one that I got 71% on. So it was like five out of seven. And then for the first three that I was talking about, it was like two mistakes out of 15. So 13 out of 15.
Got it.
Um, so here's the thing. There was actually a couple problems I wanted to go over in the sciences.
Okay.
Um, And then I definitely want to do one more car session,
Uh-huh.
probably tomorrow,
Uh-huh.
just because I want to like fully finish reviewing this full length four.
Yeah.
So yeah, are you like any opinions, I guess, on what I'm doing? Also, one other question I did want to ask you. I mean, um I have around 10 cars passages left because I was redoing all the AMC stuff.
Okay.
Um, I
All right.
mean,
Are,
sorry?
are you gonna ask if it would be a good idea to do those in like a test setting?
So actually, I wasn't. I was gonna ask. I mean, that's a great point you bring up, but no, I was gonna ask, how you think I should split those up in terms of what we were talking about, like, stopping to do stuff like a day or two ahead. Like, like should I do five tomorrow, five Wednesday, and then do nothing Thursday? Should I like leave one for Thursday? Like, what's
Yeah. So the cars that you did today, how did you do that?
so today I did like three passages and um I went over my wrong like answers.
Uh-huh.
I didn't do like blind review because at that point it was like I did a lot of blind review. I just like went over the two mistakes I got,
Got it.
and then I decided to do one more passage and I did the same thing. I went over the two mistakes.
Uh-huh. Got it. Okay. So yeah, for the rest of these, well, I would say not to do anything on Thursday,
Okay.
but for the cars passages between now and then, we can try like your strategy of being like, and I don't know if you use this for the cars passages that you just did, but I think yesterday we talked about like and again, if you don't want to do this, you don't have to do it. But like, I think we talked a little bit about like kind of like giving yourself a little pause to think about, either whatever the passage is saying or to think about whatever the question is asking. What do you think
Yeah,
about?
it was very helpful like today. I mean, obviously on the actual exam, I'm not gonna be writing like typing up the main idea, but like today when I would like type up the main idea and send it to like in the draft email, it was definitely helpful. It was helpful for sure.
Yeah, I mean more so, uh, like basically either you read a paragraph and then you give yourself like a couple of seconds to think about what that actually means. And same thing with the questions, um, to like stop, pause, make sure you know what the question is asking, and then also have like a prediction for that for what that answer is. That way you're not just looking at any because yeah, and I sent you like the email with like how like I was like timing it and stuff. Like when you were uh when you weren't thinking about it and you just read the question and you just went into the choices, you spent like 1.5 minutes to get like the wrong answer. But then when I blocked out the choices, and I made you actually like process what the question's asking and then come up with your own answer, it only took like 30 seconds. So uh so again, like you don't want to think about it in like if you are getting the thought in your head like I'm wasting time, that's you want to kind of not think about it that way because you're like what I wrote in the email was you're using time, not wasting it. You're using it so you can get the questions right. So yeah, when you saw the passage and you saw it was 7 questions long, um, it should also make sense that that should like be more important, right? Because if you understand that passage, you can get 7 questions right, whereas any other passage you can get 5 questions right. You know what I mean? So
Yeah.
I think the most important thing for you to do is to just straight up like just pause. Like pause to think,
Mhm.
right?
Okay.
Because yeah, if you're like if you're halfway through the passage and none of it's making sense, like what can you do, right? You can't really do like other than obviously like slowing down to make sure you understand what it's saying. But if you don't understand what a passage is saying and you're rushing through it, like it's not gonna turn out well.
Okay. Honestly, let's just go over a passage then.
Yeah.
Yeah. So okay, here's the thing. This passage, I got everything right.
Mhm.
It was a very interesting case, but I like didn't really understand what like I kind of understood what they were talking about, but I didn't fully understand if that makes sense. I mean, I got a 5 out of 5, so um yeah. Anyways,
Yeah, I would say if you got a 5 out of 5, you likely understood it, right? Um, but what are you're saying like even though you got a 5 out of 5 that you feel like you didn't understand it too much or something?
like I just feel like I maybe got lucky on like one or two that ended up me getting a 5 out of 5.
Eh, I mean, yeah, we can look at it, but if you got 5 out of 5, or if you like at least for the ones that you feel like you guessed, like if that makes sense, then I think you're good. I mean, we can go over it, but
Okay, well, fine. Now you're making me okay. Let's just. I know I'm being indecisive, but let's just finish full length four then.
okay.
Because honestly, time is like of the essence, and especially if I'm not gonna be doing anything on Thursday, I don't wanna like keep putting off this full length. So, okay. I actually want to go over one psych passage. Do you see it? Okay. So one thing I noticed in general in psychology, it's kind of unfortunate, but there's always one passage that I get like two or three wrong that has led me to like drop from like let's say 131 to like a 129.
Yeah,
And th this was here I got 3 out of 4 wrong. So
I see.
yeah.
So, okay, yeah.
Individuals demonstrate a variety of cognitive biases to maximize self-enhancement. Researchers investigated whether the self-enhancement gained by the tendency to favor one's in-group, so that's a buzzword, over his or her out-group, um, would influence the person's decision for selecting dispositional rather than situational causes for a target's behavior. So, yeah, I highlight like the buzzwords in-group, out-group, dispositional. Dispositional is like, I guess, personal, personality. Situational is like external. Um,
And this is all related to like the actor observer bias, the fundamental attribution error, uh, self-serving bias.
yeah. Yeah. Surveys were mailed to 398 employees of an organization who are classified as either managers or assistants. The survey was returned by 42% of the sample, 64 managers and 104 assistants. So there's more assistants that returned it than the managers.
Yeah.
Um, Survey consisted of four scenarios describing the behavior of a hypothetical target employee in a company attempting to address a common office situation. Okay. In-group and out-group bias was manipulated by alternate, alternately describing the target employee as either a manager or as an assistant. Target's behavior was described as having either a positive or negative outcome. Okay. Positive or negative outcome. The positive outcomes represent favorable behaviors such as competence and success. And then the negative outcomes um, were unfavorable behaviors such as incompetence and failure. For each scenario, participants were asked to determine whether the cause of the target's behavior was either dispositional or situational. Okay. And now for the results. So overall, the participants indicated that the cause of the target's behavior was due to dispositional factors rather than situational factors. However, results differed when group membership and the outcomes of the behaviors were taken into account. So, yeah, group membership and outcomes of behaviors. When participants made in-group judgments, managers judging a manager target or assistants judging a assistant target. Okay, so in-group judgments and the behavior outcomes were negative. Participants mainly attributed the behavior to situational factors. When participants made out-group judgments and the outcomes were positive, participants attributed the behavior to situational factors. Again,
All right, so tell me about actor observer bias.
uh, so it's when you attribute, um, like if you fail something, you attribute that to something external, while if someone else fails something, you attribute that to internal, dispositional things.
And then the other part of that?
What do you mean by that?
Uh, so you said it for negative things. Do it for positive things.
Well, I guess for positive, if you do something well, it's internal. And if something does something positive, it's external.
Good,
Someone else.
good. There you go. All right.
Okay, so this one I did get right, but we'll go over it. What concern about the study reflects problems as a result of the response rate. So yeah, uh, this was a pretty big thing. It was 42% of the sample that returned the survey, and there's a lot more assistance, like 40 more. So, okay, participants. So reflect problems. So what's the problem? Participants provided four responses to four hypothetical scenarios. Wait, I actually got this wrong. Never mind. Uh, so okay. Yeah, I got the last one right. Participants may differ from the employees who did not respond to the survey. Um, okay, so this I think I'll keep it for now. Manager and assistant participants may have interpreted the scenarios differently. So that's what I originally put. Because my thought process is that because there's a lot more there's 40 more assistants. So I mean it might skew the results a little. That's what I was thinking. And also there's less than 50% of the sample responded.
Okay. So we're being asked what could be a problem that is due to the response rate, right?
Mhm.
So if the manager, so I mean, we would imagine like the managers and assistants interpreting the scenarios differently, right?
Yes.
because if they are like if the like participant was an assistant and the target, or the scenario, the hypothetical target is also like an assistant, then it's going to be like certain effects. And then if they're managers, there'll be those effects. So like the manager and assistant participants may have interpreted the scenarios differently. I mean, that's expected, right? But we're being asked like what can be a problem due to the response rate. Now, in other words, right, that would be like saying that the difference in, like, C would be saying that the difference in the way that a manager interprets a scenario and the way that an assistant interprets a scenario is due to the response rate of 42%.
Got it. Okay, that makes sense. Yeah. So here really shouldn't have even looked at this. I should be looking at this 42% mark.
No, you're where you're looking is fine, but I think a theme from like the car stuff that we were doing was to make sure you understand what the question's asking,
Hmm.
right? Um, because like also fitting in with that theme, like if you don't know what it's asking and you don't know what to look for, you're going to be just more susceptible to these answer choices. Like you're not going to have a good reason to eliminate something or a good reason to choose something if you kind of don't have uh the understanding of what the question's asking and then your own answer. So, but yeah, they're asking what are the problems that are a result of the response rate, and the response rate was like does not very representative of like the general population, which means that you're not going to have generalizability and all that stuff. So, yeah, so this just is asking about what problem could be caused by the response rate.
Okay. Um, okay. So I would then guess that the answer would be B
Mhm. Yep.
because so basically like the other, let's say 58%, they might have had like a different, they might have like responded differently
Hmm.
and that would have changed the results.
Yep.
Okay. Yeah. Okay.
Yeah, you want to like actively think as much as possible.
Hmm.
So, um, like that also will help you anticipate things that they'll ask you. So, like when you read this and you saw, surveys were mailed to like 400 people, but 42% responded, um, that could present a problem down the line. And also, the 42 that did respond are not balanced. Like there's a lot more assistance, so that can be an issue. And if you can identify that as a possible issue or limitation, especially in psych soc, because they like to ask about like what's a limiting fact, like what's a limitation of this study design or experimental design or whatever. So yeah, you want to be looking for those things when you're reading stuff in all the sections, including cars. You want to be reading actively, you want to be thinking um as you read, right?
Hmm.
uh you don't want to do it passively if you like. Yeah, so okay, let's see. How would the pattern of attributions found in the study most likely be predicted to change during times of intense group conflict between managers and assistants. Okay. So what do we do first?
Hmm. Okay. Wonder what the question is asking. Like, think about that.
Yes.
How would the pattern of attributions found in the study most likely be predicted to change during times of intense group conflict? Yeah, this was tough. I saved this for the last second. Like, I looked over this question multiple times, did not know what to do. So it asks about the patterns of attributions. So that's referring to like the last, well, I mean, second and yeah, no, the last paragraph. And how that changed when there would be conflict.
Yeah.
And like, I don't know. Um, Recognizing the need to reduce conflict, situational attributions would be the most common. And I think I put that. Um, okay. I I don't really know. Like I really don't know. I don't remember studying anything about reducing conflict in general.
What are we being asked?
Like how it would change.
Um, like during times of conflict.
Like the attributions, how they would change during conflict.
Okay. So so far we've been talking about what main kind of phenomenon.
Like actor observer bias,
And then what's another one?
fundamental attribution error.
Oh, so sorry. I just mean also like in group, out group.
Oh, yeah.
So if there's more conflict between the two different groups, how could that influence in group versus out group preference?
I mean, you'd prefer your in group.
Okay. So it would do what to those biases?
I mean, like I guess it would make them more extreme.
Okay.
It's just like the thing is, I was also thinking, I mean, when you're in a conflicted scenario, like let's say with someone, like you're arguing with them, like what if you also attribute that to just being situational? Like you both just agreed that whatever happened was situational and neither of you was at fault, or like it was neither of your personality or whatever, and you know what I mean? I don't know.
So I think you're trying to trying to answer the question by reducing the conflict.
Mhm.
Because if in the situation that you're giving me, you guys both agree on that, it seems like you're not really having a conflict. You're reducing a conflict, right?
Huh.
So so we're being asked what happens to those biases when there's an increase in conflict. So we're trying to answer that question rather than trying to answer the question of what happens if the conflict dissipates,
Okay.
right? So what are we looking for?
But like the in-group bias would become more extreme. So this,
Yeah, how many seconds does that take?
I didn't time it, but
But my point is that
yeah.
that's how you want to do whatever you so I even did the math. If you have a high accuracy and you answer like 43 questions, you get a higher score than if you finish 53 questions with a lower accuracy. So let's say like 3 out of 5 to like 4 out of 5. Okay. So I think the biggest thing for you, like, is just to allow yourself, first of all, just to understand that you're not wasting time, you're spending it, right?
Yep.
So for this question, I know that you want to just answer it as fast as possible, but we want to answer it correctly, right? And that involves you like having to think about it because this is a critical thinking test, right? So if you're not thinking, doing it wrong. You want to be thinking. And and then yeah, and also like allow yourself to come up with your own answer because then it's just super easy because you know what to look for and pretty much that, yeah. So uh for that question, you see how A, which is what you chose, is what you were kind of explaining to me. You're saying something like when I first asked you about this question, you were saying like, what if you guys agree that it's like a situational and blah blah blah.
Mhm.
But you see how that is about like reducing the conflict,
Yeah.
being asked what if the conflict increases, what effects that will have.
Yeah.
Okay.
Based on the findings in the study, what is the most likely prediction when people make attributions for their own behavior? Okay.
What's your answer?
Um, I mean, everything was like due to situational factors. I remember that. And there was like disposition, if it was like dispositional, or no, everything was mainly dispositional, but then there was like the positive and negative was um
Uh, maybe I guess you might be thinking about like the study or something, but this is really so tell me what they're asking here.
when people like make attributions for their own behavior. So maybe it's like self-serving bias.
Yeah, right. Basically, if my own behavior is bad, it's because of external factors. If my behavior is good, because of internal things, right?
Yep.
And then vice versa. If I'm doing something that's bad, it's because of external factors.
Okay.
So what are we looking for?
Bad is external and good is internal.
Yep, that was pretty fast, right?
Mhm. Question is, why does it say like based on the study?
Because it's true based on the study as well,
Mhm.
right? But strategically, the entire test is designed to have you do what I tell you not to do, which means scroll up, which means someone would read this right away, not understanding it, and then go back to the passage like and frantically search for some type of answer that's not going to be the right answer.
Mhm.
So it's a counterintuitive thing, I know what I'm saying, which is to stop, pause, because you're running out of time. But you want to do whatever is gonna give you, the right answer. So,
Okay. A consultant is hired by an organization to break down barriers between managers and assistants by conducting a series of team building activities over a period of time. If the team building activities were successful, how would they most likely influence the attributions made by the organization's employees? People would like each other more.
okay. And how would that affect, the disposition versus whatever stuff?
I mean, people wouldn't, there would be like less active observer about, like, probably just less fundamental attribution error.
Hmm. Hmm. Yeah, you got it right before, but yeah, you got them all right now.
So, ooh,
Yeah, so try again, it's up to you, but I think we see that if you let yourself think, it's gonna be good.
yeah, I'll try. I will try. It's so like it's so hard to like keep my confidence up right now in terms of like in general, like this whole week. I'm saying my confidence is like pretty high, but like maintaining that confidence at least with the main three sections, like cars, I'm also trying.
Mhm.
But
Yeah. Now I understand that. Um, but I think that if you just trust yourself, your ability to think critically, that's going to take you take you far. But if you don't think that you can improve, or if you think that the way to improve, which is like to like slow down, is not something that you're willing to do, then that can be tough. So the middle ground, I think, would be like, first of all, so I was thinking also when you were saying that thing like 1100 BC vernacular,
yeah,
like, so that shows to me that when you're reading something that you're thinking about it. So because I was trying to think of like how to like get you to like kind of like brainwash yourself to find it interesting, but I think like if you yeah, just reading actively is going to just work well for you, and um yeah, stuff like that. Okay, so let's see.
yep.
Peptide stuff.
Yeah, so it's consisting of 9 amino acids that are partially hydrolyzed.
Hmm.
Three different tripeptides were isolated. None of the tripeptides share a common amino acid.
Hmm.
Based on the data, what is the total number of possible structures possible for the full-length peptide?
Hmm. All right, what do we do?
Um, well, there was 9 amino acids, so like the thing. Okay, so it was like basically 9 amino acids, but it's like they're like triplets because like then they're isolated, they're hydrolyzed, so it's like the chain of nine is split into three different chains of three.
Okay.
Now, that's why the rest I didn't really understand because, like, what does that mean? None of the tripeptides share common amino acids. So I guess it's like they're all 9 of them are different amino acids. Um,
Hmm.
yeah. As for the possible structures of the full-length peptide,
Hmm. Yep.
Yeah, I wasn't sure to go about this apart from just like what like writing out like 1, 2, 3, 4, 5, 6, 7, 8, 9, 1, 2, 3, 4, 5, 6, 7, 9, 8. Like
Mhm. Got it. So, yeah. So, um, possible structures for the full length peptide. Um, well, we could do like, I guess, three slots for the tripeptide, and the they consist of nine total amino acids that are, not the same. So wait a sec. Three different tripeptides. Hold on. Three different tripeptides. Okay. So we're not looking for the different configurations for the tripeptide, but or sorry, for the nine amino acid long peptide, but we are looking for the different configurations of the tripeptides. Meaning, okay, so total number of possible structures for the full length peptide. So there's three different tripeptides. So this means that in this first column or slot, let's say that there's three possibilities for the tripeptides, right?
yes.
And then since we can't repeat any, the next possible tripeptide will have out of the three, two possible different tripeptides, and then yeah, it'll be I think it'll be this. So yeah.
What? I don't get it.
So we know that there's three different tripeptides, and none of the tripeptides share a common amino acid. And how many different structures could be possible for the full length peptide? So so the three we're looking so I think this could be confusing, the nine, because if we were asked how many configurations could there be of a peptide that consists of nine amino acids, it would be like nine factorial, like huge.
That's what I thought.
Yeah. So that's going to be a huge amount, right? But they're telling us that so if we had like I'm not going to draw nine things, but something like this, and out of the and like whole peptide of the nine amino acids, there's three different types of tripeptides. So like this is one type, this is the other type, and then this is the third type.
Yeah.
So we're really looking for the different configuration possible structures for three tripeptides rather than the nine each configuration for the nine, amino acid long, like entire, peptide. So that just means then, so mainly just here, they're saying that there's three different tripeptides. And so I put these slots in so that I can have three out of the three tripeptides could be the first one. And then since we can't repeat them, yeah.
I got yeah. Okay.
Mhm.
Yeah. Next is this.
Mhm. So, um, oh yeah, it's like this is like we talked about this, right? Like with
Yeah, the basal body.
Um, the bar the bar body.
Oh, bar body.
Yeah.
What is basal body?
The basement no, basal whatever. There's a lot of things. But okay.
Oh, yeah, okay. Basal body temperature is your
Yeah.
temperature when you're fully at rest. Okay.
Metabolic rate, etc. Uh,
Yeah, yeah.
inactive X chromosome is the thing that through uh whatever um inactivation. So, okay, what applies? It does not replicate. Its chromatin structure is less condensed than that of an active X chromosome. I mean, if it's if we're able to call it an X chromosome, we get to see that it's
Nuts.
dense, right? Okay. One of the last chromosomes to replicate is highly transcribed. It shouldn't be transcribed. Um, but I guess because when it comes to, I guess it could be one of the last ones to replicate because I'm thinking of two things. I'm thinking of like in a karyotype, right? You have like the 23 um chromosomes and the inactive X chromosome is going to be on the last one, on the 23rd one. So I'm thinking of that. I'm also thinking in females, mammalian females, they have like they're like halted at meiosis, like when they're born until they go through puberty, and then it can go a little bit farther, but up until right before fertilization. So and then it doesn't completely finish until after fertilization. So yeah, so B is definitely wrong. I think D is wrong too. And well, I guess A does not replicate is going to be wrong. So C, I guess, would be the best answer. But
Okay, I'll just memorize that.
yep.
Um, do you think we'll have time to do this? Maybe like quickly?
Yep.
Okay. Because I did want to go over this one. Um, okay, Streptococcus pneumoniae is a leading cause of pneumonia. At the site of infection, innate immune cells, okay, that's a buzzword. Release toxic reaction oxygen, okay, ROS, that's a buzzword, including peroxide that damage microbial DNA. High levels of these ROS can also damage host cell DNA by causing double-strand breaks, which can be cytotoxic. I don't know if you consider this a buzzword, but like I guess I'll highlight it. DSBs activate a kinase pathway that phosphorylates histone H2AX to form um, gamma H2AX, which recruits DNA repair proteins such as 5'3BP1 to damaged area. Okay, so the double strand breaks leads to recruitment of other enzymes, and they come and phosphorylate this histone
Mhm.
and repair the DNA. Okay, as pneumonia produces a pyruvate oxidase. Okay, so pyruvate oxidase. Oxidase is a type of oxido reductase, and it catalyzes the formation of hydrogen peroxide. Okay, and that's a type of ROS. Yeah. Um, Researchers hypothesize that secreted bacterial factors, such as peroxide, are the primary cause of DSBs in host cell DNA. Okay. Researchers inoculated cell media with each of the three virulent S. pneumoniae strains to a final concentration of 1.25 times 10 to the ninth CFU per milliliters.
I hate that.
Colony forming units. Um, okay. After 7 hours during log phase growth, the media was filtered to remove the bacterial cells. Okay. Then alveolar epithelial cells were incubated in this conditioned media for 7 hours. Okay, so epithelial cell media with strains to this concentration. When they removed the bacterial cells, they added ACs. An assay was used to show localized expression of 5,3-BP1. Okay, so that's the DNA repair protein. And or Y2AX, so the histone that gets phosphorylated. And the ACs using fluorescently tagged antibodies. Okay. Conditioned media was prepared. Okay, wait. So yeah, percent cells positive. Okay, so negative control. Strain one, strain two, strain three. Negative control is something that you know is not going to lead to any result. Right. And conditioned media was prepared in experiment one. ATs were incubated in the media in the presence or absence of catalase, which decomposes H to peroxide to water. Yeah, catalase. Yeah, it would be a proportion. And then I looked at apoptotic cells. Okay, so when you would add catalase, less apoptosis. That's significant in all of them. So, okay. Which of the following explains why increasing the initial concentration of pyruvate. Okay, I mean, I got this. I don't want to misspeak, but I think I got it right. And this is Le Chatelier's principle. I feel like they test this like at least once in every exam, the last three exams.
Oh yeah, yeah. No,
It's been a freebie.
it's always gonna be a thing.
Um, so okay.
What was this? What does this positive cells thing mean again?
Positive cells.
Here,
So
y-axis here.
so
Sorry, positive for this.
yeah.
Got it.
All right. This I got, I think, wrong, or I actually just marked it. But anyways, I want to go over it. So equilibrium constant expression. So I guess that's just products over reactants.
Um, yeah, and yeah, it seems like there's no coefficients, but the coefficients would turn into the exponents, right?
Yeah.
But yeah, pretty.
Okay. So, oh wait, what? I got this wrong. Yeah. So okay, that makes sense. Why I have it marked because it shows that. Yeah. So I got this wrong. Okay. Got it. So they were testing the fact that we don't include water.
Oh, okay.
Got it.
So
So
yeah.
here's the thing that I wanted to also tell you about. I guess they didn't like put it here, but one thing I was kind of confused about is when I was looking at all these choices, they include like a solid or whatever. Like I was just confused that they're including things that normally I wouldn't see in like
Where
they
which thing is a solid?
like okay, maybe it's not a solid, but acetophosphate, like that's a protein, no?
Uh, I mean, not really, right? It's an acetyl group, which looks like that.
Hmm.
I guess connected to some phosphate like that.
Yep, yep.
But uh, so 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 yeah, it's not gonna be a solid, but the reason that we don't include solids and liquids is because they're pure substances, and if they're pure substances, their concentration is going to be 100%. So there's no way for us to, if we have a pure substance, to find out what its concentration would be, right? Like that's the reason why we don't include those, right?
But like what do you mean because they're pure substances we don't include like
Yeah. So for instance, um, like pyruvate, you can have pyruvate dissolved in water, and you get some, the moles of pyruvate over the moles of the solution, or over the volume of the solution would give you a concentration, right?
yeah.
Um, but if you just have pure water, like just straight up water, nothing else in it, then that concentration would be like kind of like a moot thing. Like you don't really makes no sense to really think of it in terms of a concentration because it's just 100% water.
Okay, so you're saying because usually we find concentration of stuff that is in water. So
That would be yeah, that would be for something that isn't water in water. That's when it won't say liquid, but it'll say aqueous,
okay.
right? Aqueous means it's dissolved in that water. But water itself, like, isn't going to be like a solute or whatever, right? So if you just have H2O liquid, it's a pure substance. We can't have a concentration value for it that isn't 100%, right? So
Mhm.
that's why we don't include it in this.
Would you be able to erase your drawings, please?
Mhm.
Thank you.
Do you have any other questions for this passage?
Yes, there's one other question.
All right, I'm gonna leave the thing on the bottom if that's okay.
Oh yeah, yeah, yeah. Sure. Okay. All right, this is next. So basically here, from what I understand with like negative controls, I mean, so I'm here. It asks what is negative control. They said that the media was filtered to remove bacterial cells, and then the AECs were in the conditioned media for 7 hours. But so I would think that it's either conditioned media without AECs or like unconditioned media with AECs. You know what I mean? Like you got to keep one thing constant.
Hmm.
Now, what are we testing for here? Um, we're testing for the amount of like expression of these two things in AECs.
Hmm.
So like I would think it's C.
Let's see. They, what do they mean when they say the conditioned media?
So it has like the strain, like the strain was in it before.
Okay. So that would help us, like, I guess, uh, that would influence something here, right?
The question is, I was wondering why did they remove the bacterial cells? Why they add the strains and then remove them?
Um, yeah, I guess I was gonna ask you a little bit about this too, like, but I guess before that, right? So they got rid of the bacterial cells, and then this became called this conditioned media. So there might be something still in there that's from the bacteria, perhaps.
Okay.
Like I was gonna ask you about like transformation,
Yeah.
where you can have, I mean, so that's where you can have like, you can kill or get rid of the bacteria or whatever. But there still could be parts of the bacteria that are in the environment that can get taken up by other cells. But yeah, I think so if we're trying to do a negative control, um, and we see in, I guess, in figure one that we have various values that uh, tell us, if this is expressed or if this is expressed, and you can see that those produce uh, measurable positive cell percentages, right? That these things, right, could affect that, right?
Mhm.
So, so what we want is we want some type of negative control in which we would see what would happen if no type of like let's say treatment was done. Um, so yeah, so I guess if we're between unconditioned media with AECs, then conditioned media without AECs. Um, so I think this conditioned media part is what makes it not a control because like the strain one, strain two, and strain three were, you know, they were put together and then sure they removed him, but clearly in the figures we see that even when they remove them, they still have these measurable, the effects. So the conditioned media is not going to work to as a control, a negative control, because it already has these, strains or pieces of the strains left over. Okay.
Which experiment was best linked the result of experiments 1 and 2 to show that S. pneumoniae can cause apoptosis of host cells by contributing to DSBs?
Mm-hmm.
So experiment 1 measured the like amount of the 5,3-pp1 and the white gamma H2AX using antibodies. Okay. This measured apoptosis. So we know H2O2, um, like, yeah, here. Talks about catalase decomposes peroxide to water, and we know that peroxide causes damage to DNA. So like should probably be something with double strand breaks.
Yeah.
I think if we determine other factors, like that literally does not do anything. That makes us like farther away than closer
Yeah.
to like comparing the two. Levels H2O2. Um, I mean, we're kind of already measured that indirectly here. Um, and determine if it's produced by other strains. Like we're no, because we want to show that that S. pneumoniae is the one that causes apoptosis. So yeah, I mean, I'm leaning towards D.
Well, the different strains are still of S. pneumonia.
Yeah, true, 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 true. But
So,
like,
uh-huh.
yeah, but what's the point? Like, we're already looking at 1, 2, and 3. Adding additional strains we would need another experiment even on top of that.
We don't have to add any strains, but we can examine like so if we know that double strand breaks can cause apoptosis, and we know from passage that double strand breaks can happen when you have ROS or eight in hydrogen peroxide. Um, that can help.
Yeah, I mean, I knew that too,
Yeah.
but yeah, like Ross leads DSBs and apoptosis.
Okay. So so yeah, we're seeing what how we can link the result of experiment two to experiment one. Experiment two talked about the connection between H2O2, the peroxide, and the apoptosis,
Hmm.
right? And experiment one talked about the peroxide yield
Yeah.
leading to ESBs, which can lead to apoptosis. So we want to know, like, yeah, how can we just connect these?
I'm guessing it would be D.
Um, that so okay. Um, yeah. So what we're trying to show here is for that. And so I guess number to measure the number of double strand breaks that occur in AECS cultured with each strain versus measured levels of H2O2 produced by each strain. And we're trying to link these two together. And it seems like the link that they can both have is the amount of that peroxide, right?
Yeah. Is there any way we could sneak in one discrete?
Yeah, yeah, of course.
Okay, okay. Your last one, because this is like I think the last problem I have. In the whole exam, it is this one.
Oh, great. All right. So, all right. White eyes are recessive to red eyes, and yellow bodies are recessive to dark bodies. Um, it's going to be a dihybrid cross, right? Okay. But both genes are on the X chromosome. The genotype of a red-eyed recessive, dark-bodied dominant female XX could be determined by mating it with a male XY that has which of the following phenotypes? Okay. So, oh, I guess this isn't that bad. All right, as bad as I thought it was going to be. So hold on. White eyes are recessive to red. Okay, so the female is red-eyed, meaning it's dominant and also has dark body, which is dominant, right? So
Mhm.
the female has both of these dominant alleles, right?
Yeah.
And then the male here, um, oh, so yeah, so we're gonna try to figure out the genotype of the I guess the female by mating it with a male with a certain type of phenotype. So if the female has the dominant red eye, if the male also had the dominant red eye, we won't be able to tell if the female is because let's say heterozygous for dominant is going to give you uh the same phenotype as this, right?
Okay. Yeah. Yeah.
So we don't want the male to have the dominant red eye because we won't be able to tell which genotype would be between these.
Hmm.
So I think those could be out. And then the female had dark body as well, and for the same reasoning, um, we would want our male to not have a dark body, have the recessive version of it. So D.
Okay.
Yeah. Yeah, that wasn't that bad. Looked bad.
Yeah. Okay. All
Um,
right.
yes. So in terms of your cars practice, read actively, constantly as you read, think like so imagine that this is like your best friend who's like, I just spent all the like I spent the last 3 years writing this dissertation. Can you please check it out?
Yeah.
You're gonna you may not want to, but you're gonna be like, okay, like I'll listen. I'll do it. I'll do it. So there's that. So like read actively as much as you can. Um and pause if you need uh to think. If you are not thinking, like catch yourself. If you catch yourself not thinking, like if you're just like reading the question and then reading through the question choices and spending that much time doing that, uh best bet is to just pause and think about it. And I think we've kind of proven, like you still may be skeptical, but I think we've kind of proven that when you just simply like when I block everything out and you just come up with your own answer, you pretty much instantly then find that right answer. This time you save from that pause is weeks like dividends. You have a lot better results that way. So you're like a trillion% capable of all this stuff, right? So I think that you're at the stage where it's less adding things to your knowledge and more removing things that would otherwise stifle you.
Yeah.
So I would just say to yeah, so read with a purpose, like constantly be like instead of passively skimming, right? Always try to anticipate the author's intent. Um and like try this out when it comes to like Wednesday. I'm gonna give you or Thursday, like I'm gonna give you like because I've also been working on like okay, like what like break glass in case of emergency, like everything's like, running out of time, what to do then. I have some of that like that I'm working on that I'll let you know like of what to do, like worst case scenario wise. But yeah, until then, I would say just read actively, try to anticipate things that they'll ask. Um, Also, once you could start seeing like yesterday, we saw that like 3 paragraphs in, we saw like a repeated argument, and that can help us especially if you're running out of time, because if we see that repeated argument in the first 3 paragraphs, like it's going to constitute a main idea, and they might talk about other things later, but if you're completely like strapped for time, right? You can kind of whenever you see those repeated ideas, think about them as possible main ideas. Um, and always, yeah, just try pausing and making sure you understand what the question's asking, and then try to come up with your own answer. And the other thing is just I guess don't think of it as wasting time but spending it.
Mhm.
Okay? Because uh you want to do that. So I think that's all you need basically. That's the only thing. Like I think just doing that will significantly improve your score. Okay.
Mhm.
All right.
Um,
Yep.
okay. And you wouldn't suggest doing like a whole section of 9 passages? Like you wouldn't suggest that? I mean, you brought it up yourself. I was just wondering why you brought it up. Like,
Um. Oh, this is like when we were talking about earlier in the.
we were talking about it like at the beginning of the session, like what I should do with these like 10, 11 passages I have left.
Yeah. Yeah. I'd say no more doing it. Well, okay. If the question is doing it timed versus not timed, is that the question?
Not really. It was just I'm doing everything timed. It was the question was just if I should do a section of 9 passages.
Oh, in a row like that.
Yeah.
Okay. Um, and if you did that, you would do that when?
Tomorrow.
And we do it tomorrow, um, and I guess that would be if you do it tomorrow, that would be the last like actual like practice passages you'll be doing, right?
For cars, I mean, yeah, basically, I'd have like one left, which I would maybe do Wednesday, but it's just like everything I'm doing now is really trying to like baby myself. I don't know, that really doesn't make sense, but like
No,
make myself think that I got it. Like
yeah,
not
yeah, yeah.
like really not d destroy this any more confidence, you know? Like
All right. I don't uh care if you do it timed or untimed. I will say that's up to you. Only thing I want you to practice is reading actively, um, pausing, thinking about either what the paragraph said, what the passage said, what the question sem is asking, and then coming up with your own answers, and then looking for that answer. However you do that, whether it's timed or not timed, um, I don't think I personally don't care about that, but I want you to do those other things. I want you to not feel like you're wasting time and to emphasize efficiency, which is rather than just finishing as fast as possible, having that balance between this is the rate I must go at to get this percent of the questions right. That will always be preferable. So what not to do is like if you're halfway through a passage and you're not understanding it, like anything, um, it might be best to I'll come up with a good answer to that. So just making it really, really short. So basically, for the cards that you're going to be doing, just yeah, actively read. Always be thinking like, okay, this is cool, like, but that's a little weird, why they're saying it this and blah blah blah. Actively read, learn to anticipate things that are said as you read things, and let yourself pause to think about what the question's asking. I think that's going to be a big part. Okay.
okay.
Yeah. All right.
What
So
was the total?
yeah, let's see. 94.
Okay.
Okay, awesome. So I will see you tomorrow. Okay.
Let's shoot.
All right. Take care.